All right, so in the interest of time, we're going to go ahead and move on. Um, I kind of overheard some of y'all saying, why is this packet so big? Right? There's a lot in this one, but hopefully you'll see that a lot of it is just kind of short little answers. Okay, so we are now going to start thinking about some of these catalytic mechanisms. So how do enzymes catalyze the reactions? How do they carry out those features we talked about, dampening harsh conditions, decreasing activation energies? Okay, and we're going to think about this from the perspective more of like organic chemists. Okay, but hopefully I'm going to point out to you some themes in biochemistry that are very different than what you learned in organic, right? One of the biggest ones is remembering that general acid-base catalysis is everywhere. Okay, what we mean by general acid-base catalysis is pretty straightforward and pretty simple. It's that if you need to have an acid or a base, it's there. If you need to deprotonate something for a reaction to work within the constraints of the enzyme active site, it can happen. Okay, that's what we mean by general acid, general base catalysis. And you are very, very commonly going to see me or see other biochemists using this kind of like general system where you have B with a lone pair. This is a general base, right? By using this approach, you're saying, I understand that I don't know what the active site looks like, but I also know that for this reaction to work, I need a general base to be able to deprotonate water. So I have this general base, it reaches out, it pulls the proton from water, that creates hydroxide, and hydroxide's a great nucleophile, right? So using this idea that you have a general base as a B, the general acid we usually list as an A with a proton bonded to it, okay? This could be a general acid. If you have the need for a general acid, right? If you need to protonate something, then you have a general acid. Whatever you need to protonate reaches out, pulls the proton off your general acid, and you then generate a general base and a protonated substrate, okay? We use this generically because truly general acid, general base catalysis is part of effectively every single enzyme mechanism there is, right? Protons flow around in biology all the time. So don't get hung up, don't get stranded on this idea that I need to protonate something. Just do it. It happens. It's there. Okay, and we'll look at an example. Covalent catalysis is a form of catalysis where there's actually a covalent intermediate that forms between the enzyme and the substrate. Okay, and the big mechanism we're going to talk about today, assuming we have time, is serine proteases. Okay, and there we're going to see an example of covalent catalysis. There's a serine side chain that forms a covalent bond between the enzyme and the substrate polypeptide chain that it's hydrolyzing. Okay, that's covalent catalysis. So if at any point you see a covalent bond between an enzyme, like a side chain in the active site, and your substrate, that's covalent catalysis. Catalysis by proximity. This is the idea that if you have two substrates, one of the things that slows down reactions outside of a cell is having low concentrations, right? For a reaction to happen, two things have to come together. They have to bump together. And not just bump into each other, they have to bump into each other in a way that they're oriented properly that they can react. This is the idea of catalysis by proximity. Enzymes are really good at binding substrates, putting them close together, and making sure they're oriented in a way that things can actually react. Okay? Oftentimes, outside of the active site of an enzyme, this wouldn't happen because then it's very random, right? It's random chance they bump into each other correctly. But in the active site, that happens. Oftentimes, it's helped a lot by metal ions. Okay, and we're going to see some examples of this. Anytime a metal ion is involved, whether it's involved for electron transfer, or whether it's involved to help orient substrates, or whether it's involved to help activate a nucleophile, we'll see examples of all these today. That's an example of metal ion catalysis. Okay, and then the final one is preferential transition state stabilization. And this, lots of words, but it boils down to this idea, is the transition state is high energy. The enzyme creates an environment that stabilizes that high energy transition state. And by doing so, it decreases the activation energy. That increases the rate of the reaction. Okay? 5 types of catalysis. Lots of enzymes will use all 5 of these. All right, so here's one really good example, ribonuclease A. This is an enzyme that's present everywhere, right? So if any of you have worked with RNA in the past, you know how careful you have to be about making sure things are RNase free. It's because this enzyme is everywhere and it is robust, it is stable. You can boil it, you can autoclave it. It doesn't care. It just sticks around and will keep on chewing up RNA, right? So it's a really, really beast of an enzyme. Okay, so the enzyme that it catalyzes is it uses water to hydrolyze the phosphodiester backbone of RNA. Okay, so here we notice that we have a polymer of RNA, and we know it's RNA because we have a 2' hydroxyl group. We're adding water, and what we notice here is that the backbone now has been hydrolyzed. We have added water across the phosphodiester backbone. Okay, this is the enzyme. So the enzyme catalyzes this hydrolysis reaction. What I want you to note, and hopefully you can see here, is that in the active site, there are 2 histidines that are positioned adjacent to each other. They're positioned directly across this channel, this groove where RNA binds. So the idea is RNA binds, and when it does, whichever of the phosphodester backbone is positioned between these 2 histidines, that's the bond that gets cleaved. So in this image, let's see if I can do this real well here. So in this image, if this is the chain right here, then it would be cut right there between those 2 histidines. That's the active site. Those 2 histidines are incredibly important because they serve as a general acid and a general base. Okay, here's another kind of cool example. So tryptophan synthase. This is an enzyme that ultimately takes this intermediate, this indole intermediate, and it converts it to the amino acid we know of as tryptophan. Okay, we won't really get into the chemistry because it's actually fairly complicated, but one really cool thing about this enzyme is it catalyzes 2 reactions. And the intermediate that's formed here, indole, we don't want it to escape, right? So if indole were to escape into the solvent, it would diffuse away. It would do a couple of things. Number one, it would aggressively slow down the reaction, but also it's reactive enough that it would find other things to react with. So the idea here is there's 2 active sites and they're connected by this substrate channel. So in the first active site, it's the 3-glyceraldehyde phosphate active site. So it hydrolyzes this bond, it releases glycerol 3-phosphate, and it produces the indole. The indole will actually diffuse across this channel within the enzyme. It never leaves the enzyme. It's actually closed off and it can't leave. It diffuses through this channel all the way to the other active site, the beta subunit, which is the tryptophan synthase active site. So here, this is where serine will come in. It'll bind, and then we'll make tryptophan. Okay, so again, we're not going to talk about how this chemistry happens. I just want you to appreciate the complexity of this and just how things have to line up perfectly for something like this to be able to evolve. Okay, we're going to look at poly-A polymerase in pretty good detail to really kind of understand how some enzymes work. So the things that you should be looking for as you're thinking about how enzymes work. So poly-A polymerase is kind of the reverse of ribonuclease A, right? Whereas ribonuclease A hydrolyzes the phosphodiester backbone, bless you. Poly-A polymerase will synthesize the phosphodiester backbone, okay? And it's specifically looking for adenosines. So things that we need a polymer channel, right? This polymer channel should recognize the phosphodiester backbone of RNA. And then it wants to add adenosine onto the 3' end of it. So not only do we need a substrate binding channel for our RNA polymer, we also need to specifically be able to recognize ATP. We want it to be RNA ATP, not DNA ATP, right? Not DATP. We want it to specifically recognize ATP, not GTP, or TTP, or UTP, or CTP. It has to be ATP. And then it has to make sure that the proper part of ATP is positioned appropriately at the end of the polymer channel so that we can actually add on one more nucleic acid. All right, so hopefully what you can see here is that the polymer channel is kind of obvious, right? It's this kind of, like in some biology courses in high school biology classes, you think about enzymes as Pac-Man, right? And this enzyme is kind of a good example of that Pac-Man analogy. Is you look at it, it really kind of looks like it can chomp on things, right? There's this really obvious binding cleft. But what's notable about this binding cleft is look at the positioning of these blue, right? This blue is an NH. So that NH, right, just think about you have nitrogen and a hydrogen, partially negative, partially positive. So every time you see this little blue dot, there is a positive dipole. And if you were to measure that, you would see that the positioning of that blue dot is almost exactly what we would need for the space between phosphates in the phosphodiester backbone of RNA. So there'd be a nucleic acid position here with the phosphate interacting with that blue dot. The next one would interact here with that one, and that one, and that one, right? So there's a very, very clear channel where the RNA polymer can bind to, make favorable but non-specific contacts, and it leads up into this groove right here. And I don't know how you can see it, but right here, that's actually ATP bound. Okay, those orange, that's those are the phosphates of the phosphorus atoms of the phosphate. We zoom in there, and what we see is the way that ATP is bound is first we make some specific interactions between the amino group on adenosine, right? Remember, adenosine has the amine up. So this amine position here makes a very specific hydrogen bond with this aspartic acid in the active site. That's a really important hydrogen bond because this makes sure that we can recognize adenosine, not guanine, because guanine would have a carbonyl here, which wouldn't make the proper hydrogen bond. So we can recognize adenosine, not guanine. Additionally, this interaction here between this nitrogen and this magnesium ion is important because it ensures that we're looking for purines, not pyrimidines. If this were a pyrimidine, this would be a 5-membered ring. It would be too big to bind here because that magnesium would exclude it. The magnesium is positioned in a way where it will not let a 6-membered ring bind here. So the positioning of this magnesium ensures that we're recognizing a purine, not a pyrimidine. So we immediately get rid of CTP, TTP, and UTP. And the positioning of this hydrogen bond acceptor ensures that we're looking for a hydrogen bond donor, adenosine, not a hydrogen bond acceptor like guanine. Okay? This other magnesium ion you see is interacting specifically with our phosphodiester backbone of ATP. Okay, and this is really important to make sure that the gamma phosphate, this gamma phosphorus right here, is positioned in a way where our incoming polymer chain can be a good nucleophile. It positions the ATP, positions the phosphates properly for the right phosphate to be the electrophile. Questions? And I understand that if you're not used to looking at these things, it may look really foreign. So if it's, please ask, please go back, the slides are posted, look at it. If you look closely, things start to make sense, but sometimes you just have to look at it longer. Okay, so in this reaction, right? So the idea here is that we have the 3' hydroxyl of the growing polymer chain, and we have diphosphate, oxygen, phosphorus, oxygen, and here's the rest of ATP. All right, so the idea here is that we need this 3' hydroxyl group to be the nucleophile. It's going to attack this alpha phosphorus as the electrophile. So if this enzyme works properly, it needs to be able to do two things. One, well, maybe more than two, right? It needs to make sure that these are oriented properly, which we kind of talked about how it does that. It needs to create a good nucleophile from this 3' hydroxyl group, and it needs to make sure that we have a good leaving group, okay? Pyrophosphate diphosphate is a good leaving group, okay? But it can actually get even better because of this structured water. This structured water makes sure that it can readily react with this oxygen, accepting a proton as it leaves, okay? Does that make sense? Kind of not. If that part of itself doesn't make sense, I'm okay with it. The general idea is that it's positioning things properly and ensuring that the chemistry can happen in a simple way. All right, here's the more important one though, is that, so here again, so here's our alpha phosphorus. Here up here, this is our ribose ring. So this right here is the 5' hydroxyl group, and this right here is the 3' hydroxyl that's the nucleophile for the reaction. So this is the RNA polymer that we're adding ATP onto. This is the 3' hydroxyl. This needs to be the nucleophile, right? I mentioned that, and hopefully you know that deprotonating alcohols is not easy, right? Abstracting a proton from an alcohol is not always the easiest thing, and alcohols themselves aren't good nucleophiles. So one big role of enzymes that use alcohols as nucleophiles is to activate this oxygen to be a nucleophile, which means either deprotonating it or weakening this OH bond. Okay? This enzyme does it by positioning these two aspartic acids such that they make a hydrogen bond with the 3' hydroxyl. That's pulling the hydrogen away from that oxygen, deprotonating it, making a good nucleophile so that it can then attack at the alpha phosphorus. That's the big take-home message. Like if you look at this and you're lost, that's okay. I hope it makes sense. But if you look at it, you're lost. What the most important thing to recognize is that the enzyme provides a general base. That general base is this aspartic acid. It's positioned right by the nucleophilic oxygen so that it can reach out, deprotonate it, make that oxygen, that 3' hydroxyl group, a good nucleophile by deprotonating the oxygen. That's the important thing. Because that's there, this is a good nucleophile. We have an excellent electrophile, and the enzyme positions them close to each other. So the enzyme provides the perfect framework for this reaction. It allows the 3' hydroxyl to be a good nucleophile because it has a general base positioned at the right spot. It makes sure the nucleophile and electrophile are positioned close to each other. Right? Here's the 3' hydroxyl. Here's our alpha phosphorus. Make sense? If not, say so, otherwise I'm moving on. Everybody got it? Okay. All right. Other examples of general acid, general base catalysis. So this one is an isomerase, right? So the idea is that we are going from the alpha, or I guess it's going from the beta to the alpha configuration of this carbohydrate. Okay? So here we have an OH. Here we have an aldehyde. So the idea is we need to be able to extract the proton from this oxygen, move the electrons down here so that we can then create the alcohol. Right, so we're kind of like looking at the structural differences between the reactant and our intermediate. We have an OH, we have an aldehyde. We have an OH, we have this oxygen as part of a bond, right, as an ether linkage. So kind of what we want to be able to do is show just generally how a general base could be positioned here. It can reach out, take that proton, electrons from this bond move up to make the double bond. We have a general acid positioned that can provide a proton so that we end up with our OH here this hydrogen goes right here, and this hydrogen ends up as part of the base. Okay? So if we were to look at the active site of this enzyme, we would expect there to be a general acid and a general base positioned so that one of them can abstract the proton, one of them can donate a proton. We go through the intermediate where we have our linear aldehyde, and then it closes back up. Now this one serves as the general base. It pulls off that proton, etc. Okay, I won't go through the rest of it. What I really want you to focus on and recognize here is that a general acid and a general base is required. Okay, all right, so here is one last example, and then I'll give you all the rest of the time to work on the activity. Okay, so here's a really kind of cool one. So does anybody know what carbonic anhydrase does? So it's the enzyme that catalyzes the conversion of carbon dioxide to carbonic acid, right? So if you think about the relevance of this, when we breathe in oxygen, we breathe out carbon dioxide, right? But you can't move carbon dioxide through your blood, right? You move carbon dioxide through your blood in the form of carbonic acid or carbonate, okay? This enzyme is the only thing responsible for very rapidly converting carbon dioxide to carbonic acid, which can freely diffuse through our blood. And then at your lung, convert the bicarbonate back to carbon dioxide so you can breathe it out. And it's fast. It's really fast. And it's kind of cool because depending upon which isoform you have of it will dictate which direction is going to be favored. But really the way this works is that you have this histidine, which serves as a general base. It's linked up to a zinc ion, a zinc cofactor, positioned in the active site, and there are 3 structured waters. So the general idea here is that this histidine serves as a general base. It deprotonates one water, which then in turn deprotonates another water, which in turn deprotonates another, ultimately generating a hydroxide bound to this zinc. The hydroxide is then the nucleophile, which will attack at this carbon, creating carbonic acid. And then the reverse can happen also, right? So in the reverse, you have a water which you basically, these electrons move down, releases hydroxide, and then all this moves in reverse. Okay, so this is just one more example that your book shows of how acid-base catalysis works. Okay, a general base linked to the rest of the active site by structured waters in the active site of the enzyme. Deprotonate hydroxide, give you hydroxide. The hydroxide is in the nucleophile, which can attack carbon dioxide to make carbonic acid. Okay? We could go through hundreds of examples. We won't, but we could. In the interest of time, I'm not going to go into this. I do have a video posted online that I encourage you to watch because the last part of your activity, if you look starting on page, what is this? The 5th page, the rest of this is really serine proteases. Okay, and the way that I set it up is that I don't necessarily need you to be able to push the arrows but I do want you to be able to look at the steps and describe the chemistry. Okay, so on this second to last page, I guess third to last page, part E, it shows you step by step the chemistry is happening. So I want you to be able to look at this, kind of think about what's happening, and ultimately explain it in your own words. And if you're able to do that, then you can do the chemistry. Okay? To reinforce, I have a video online posted already that goes through this step by step for you and with you. Okay? So if looking at it doesn't make sense and you can't see it, then watch the video and things are color coded and I think it's a lot easier. But just generally speaking, what happens here is that this is ultimately a hydrolase. It uses water to hydrolyze a peptide bond. So we are adding water across this bond. We call this our sessile bond. It's the one that gets broken. The only way of figuring out which one of them gets broken is what is positioned right here. So the amino acid side chain just before that sessile bond dictates what peptide bond gets hydrolyzed, okay? And the identity of this is recognized by specific enzymes. We talked about this at the beginning of the class, how trypsin, chymotrypsin, and elastase all catalyze identical reactions. And the only way they're able to differentiate which peptide bond to hydrolyze is what's present right here. If this is long and positive, like lysine or arginine, it hydrolyzes. Trypsin hydrolyzes. If it's big and bulky, like tyrosine or phenylalanine or isoleucine, chymotrypsin will recognize it and hydrolyze it. If it's smaller, then elastase will. Okay? And I don't want to babble on more because I will. I just want to stop and give you a chance to work through some of your activity.